Hello, everybody. Welcome to the next this Carney Conversations, coming to you from myself, Diane Lipscombe, and I'm here together with Chris Moore. We're going to be talking with Stephen Salloway and Ashley Webb on the topic of Alzheimer's research from bench to bedside. And so I just wanted to say a couple of quick introductory things as everyone is signing on here. First of all, we really, for those of you who joined us before, we really encourage you to enter any questions that you have in the Q&A function at the bottom of your screen. That really keeps the discussion lively, and then you'll hear less of the questions from Chris and I, more from the audience. Always much better. And I'm going to do a quick introduction to our guests before we head into posing some questions to them. So again, thanks for joining us. It's really, this has been one of our most fun things to do over this time period, to just engage with our colleagues in a number of conversations about the brain. It's an incredibly fascinating organ. And actually, this month is in fact the National Alzheimer's Disease Awareness Month. And I just wanted to do a little shout out to Kelsey Babcock, who's a neuroscience graduate student. And they've taken over our Twitter and Instagram accounts for today. And so she's going to actually be also sharing some of her work that she's been doing in Ashley Webb's lab. So thanks for doing that, Kelsey, out there. But this is a discussion that's touching all of us. Every one of us has been affected in either directly through family members or friends, and seen them suffering from Alzheimer's disease or a related dementia. And we all know someone who's died with that disease. And right now, it's probably about 6 million people in the United States alone who have been diagnosed with Alzheimer's disease or a related dementia. And just the terrifying statistics is that that number is going to double in the next 30 years. We know that, I think there's no dispute actually over this assertion that good, solid, rigorous, creative, innovative research is going to lead us to new cures and treatments. And so that's why we really wanted to combine talking to Ashley, who's doing really at the fundamental level of basic research, thinking about aging and what happens to neurons during aging. And then also talking to Steve Salloway, who's been a huge figure in the area of Alzheimer's disease for many years. And so I'm just going to introduce them individually and then jump into asking them questions. So Steve Salloway is a medical doctor. He is a leading national figure in the area of Alzheimer's disease in diagnosis, treatment, care. And he has run a number of really important clinical trials in Alzheimer's disease. He's the Martin M. Zucker Professor of Psychiatry and Human Behavior and Professor of Neurology at the Warren Alpert Medical School at Brown. And Steve directs the Memory and Aging Program at Butler Hospital. Steve has made the US News and World Report's top doctors in the United States, has been recognized year after year after year for his exemplary teaching. And to many of you, Steve is really pretty well known and a very recognized figure across the state and beyond for his incredible advocacy for Alzheimer's disease and dementia research. So Steve, thanks for joining us.
Thank you.
Ashley Webb is the Richard and Edna Salomon Assistant Professor of Molecular Biology, Cell Biology, and Biochemistry at Brown. And her lab is funded by a number of research grants. And she was a past recipient of the Glenn Award for Research in Biological Mechanisms of Aging. While at Stanford during her postdoctoral training, Ashley became really interested in the underlying mechanisms that are involved in programming neural stem cells and exploring their importance during aging. Neural stem cells are progenitors of the close to 100 billion, maybe 83 billion neurons, depends who's counting, in the human brain. And Ashley researches basically, she's going to explain it much better than I will. but determining underlying mechanisms are molecules that function as kind of switches to clean out damaged stem cells to maintain their quality. It's a really important area of research with incredibly interesting implications for thinking about how to treat age-related degenerative diseases. Great. So Ashley, thanks for joining us too.
Thank you.
All right, so onward. Steve, I'm going to go ahead and start with asking you a couple of questions. And just wanted to reiterate the fact that, you, as many of us, have had relatives with Alzheimer's disease and dementia. I read somewhere that your grandmother, Lena Salloway, had dementia. So this hit really close to home for you as you were growing up. And you know the impact on families and their friends and caregivers. So I just kind of wanted you to give us a sense of your day, right? Most of us internalize this and think about the impact on our families, but there you are every day working with families, and you've just been such a tremendous advocate for their involvement and their willingness to be involved in clinical trials. I've heard you start off by thanking them and the patients that you're treating and the families that you work with, but can you give us some sense of what is your day like in the clinic?
Well, first of all, thanks, Diane and Chris, for including me and including Ashley. And it's great to have the bench to bedside. That's the whole goal here, is really to bring treatments to people that need them. And among the diseases, Alzheimer's is among the greatest needs for breakthroughs. And thanks for mentioning my grandmother, a blessed memory. She did influence me a great deal because I was at a formative age when she developed dementia, and she lived a long time, about 15 years with dementia. So it was a long course, and she lived close to, first she was staying in my room for a short time. So I really saw up close and personal as a boy what the impact is on the individual and the family. And then she was in a nursing home for many years right down the street from us. So I saw her through all stages of her illness. So that definitely sensitized me as I was trying to decide, what I wanted to do with my career. And certainly Alzheimer's there's just such an urgent need. And it's definitely made me a better doctor, just having that awareness. And so I'm very passionate about this as a physician scientist, but I'm passionate about this as a human being because I know really what it means to families and the fear associated with this disease and the impact. So in terms of a day, one real fun thing about my job is I get to integrate clinical work and research. So we try to blend that as much as possible, so we can be as productive as possible. So I spend some of my time treating patients and their families, teaching medical students and residents, about Alzheimer's and other neurologic problems. I work with a really talented group of young investigators. And so part of my time is spent helping them plan their research, launching their careers. That's very, very important that we get the next generation of leaders and as you said, really creative science to bring that to bear from many fields, because Alzheimer's is a complicated disease. Increasingly in the COVID era, we spend a lot of time on Zoom like we are today, which cuts down on travel, but it actually makes it easier. Yesterday, I had 2 or 3 Zoom calls at the same time. I'm a little old for that kind of multitasking, but I spent a lot of time with colleagues here at Brown and around the world planning research studies, analyzing data. And so it actually Zoom, the virtual nature makes it easier to do that than it was previously. And then a big part of what we're doing today is media interviews and working with our outreach team to get the word out, to increase awareness, because awareness is critical, first of all, to stimulate interest, to overcome the stigma, and to lower the barriers so people feel comfortable participating in research. And it's really going to take a really large number of people, from people who are cognitively normal, but maybe at risk of Alzheimer's through people who have different stages of cognitive impairment to make a difference. And finally, these days, some time is spent about COVID and dealing with safety issues related to COVID. And right now with this surge, it's challenging. You know, we've been really lucky up till now. We've remained open and our treatment trials have stayed open. But, you know, now the rate is going up. So it you know, the risk benefit, you just have to be super careful about that.
Great, thank you, Steve. Yeah, it is. So thanks for bringing us into your world. And you already mentioned, you know, what motivated you to go into this field and start to really try to get to the details of what is actually happening during this incredibly, you know, slow but degenerative disorder. So can you tell me, maybe before I flip over to Ashley, what is there like this moment? I remember moments where in the lab and just like saw this result, and never in a million years could I have expected this. I wouldn't have ever predicted it, which is always much better than anything that we could all, we mere mortals can like just basically predict what's going. Is there something that you can tell us? It's like, oh, I remember this moment and this is what keeps me going. And this is like completely inspirational. It's like, do you have one of those? Well,
I think there are three things that sparked my interest. And you already mentioned one in your introduction, was two of them actually, is just curiosity about the brain. What a fascinating organ it is. So that definitely, you know, stimulated me. I wanted to make a difference against a major brain disease, and Alzheimer's, just I think with the experience with my grandmother, you know, influenced me in that direction. What keeps me going is really, and you mentioned this too in your introduction, is working with so many inspiring study participants. People who put themselves on the line, sometimes first in the world to get a medicine. And we've been fortunate to have some of those moments here in our memory program. I mean, that's just amazing. People on their deathbed and their last moments of life agreeing to have a PET scan so we can have a new way to see the pathology of Alzheimer's disease safely and not wait till someone passes away. And working with a talented multidisciplinary team to really carry out our mission of making breakthroughs for Alzheimer's. So that's what motivates me on a daily basis. In terms of, you know, aha kind of moments or special moments, I mean, one we're experiencing right now, and Diane, you're so central to this, everybody actually, Ashley and John and Chris, is the maturity of the Alzheimer's research field. I didn't know that it would get to this point. We now have a national plan to fight Alzheimer's that's funded by Congress. We have substantially more funding for the NIH. We still have a ways to go, but we've come a long way. And Brown is all geared up from the president on down and highly motivated, recognizing this problem, that Brown can really make a big impact. And, you know, and you and I have talked about this, that it has exceeded whatever I thought we were. I could see it coming, but I didn't realize we would come with this type of momentum and this type of support that is being generated right now. And so that's terrific. And there have been a few breakthrough moments. We're still waiting on the therapeutic side, and we have some really good options, some promising developments, but where we've had the most progress is really in biomarkers, is being able to see the pathology of Alzheimer's. And so, you know, when Dr. Alzheimer described this 100 years ago, he had to wait till someone passed away to see the pathology, the plaques and the tangles and the neuronal loss. But now we're getting really good at seeing that safely with brain imaging tests, very soon with blood tests that really predict the same pathology. And so it's just been great to be part of that. And there have been a few real highlights in 2012, and maybe I'll talk about it in a future question, but we had a paper in the New England Journal of Medicine about what changes first in Alzheimer's disease and going back, you know, 20 years and more. And that was really a landmark paper. It was a multi-center collaborative, which I was excited to be part of. And then in 2016, we had a paper that was on the cover of Nature showing that we could remove amyloid plaques and slow down memory loss with an antibody. That was awesome to see that on the cover. Just didn't think I'd, see that in my career. And so it means that we're getting there. And then the next final thing is just working with people like Ashley and the kinds of things that she's doing with Justin Fallon, and what John Sedovy is doing here, what two Brown undergraduates did in developing their own company, and then having a landmark paper just a month ago in the New England Journal of Medicine about ALS. I mean, there's just great stuff going on. So all of these things have exceeded my expectations.
Thanks, Steve. And your generous spirit always comes across whenever you ask a question. It's what other people have been able to do, which is really.
Well, it's true. This is
amazing.
this is mission-driven work, but it's not my mission. It's a really,
Yeah.
I know. It's a real community, and it's great to be surrounded by so many talented people who are contributing in their own way.
So you gave me this perfect opportunity to move to talk to Ashley. Then I'll let Chris jump in. I'm sure that questions will begin to come from the audience as well. But Ashley, Steve was talking about this, the incredible importance, and I think this is true for many fields, but so incredibly important for the research in Alzheimer's disease, which has not been, Steve is right. It's just an incredible gift that we've got a lot of, never enough funding, but a good amount of funding now going. This realization that funding for research and enabling research to happen is going to be the path forward to find new cures and treatments. So, you came into this field, probably wasn't your original plan, but can you tell us a little bit about, what led you to start to think about the importance of stem cells, neural stem cells in the aging process, and how that led you to really be thinking a lot more specifically about Alzheimer's disease and neurodegeneration?
Yeah, absolutely. And so first of all, I'd also like to thank Diane and Chris for having us. It's great to be here with Steve and part of this conversation. So first of all, in terms of being in just the aging field in general, I think, it's just very well known now that aging is the major risk factor for Alzheimer's disease. For late onset Alzheimer's disease. It's not a disease that happens in the young brain. It happens to the elderly. And we still don't know exactly why that is. And so I'd really like to be a part of helping us to understand the mechanisms that are responsible for Alzheimer's disease developing the aged brain. And so, as you mentioned, a major area of research in my lab is on the mechanisms by which new neurons form in the adult brain. And this has actually been sort of a controversial area over the years. And there have been some papers that initially said there's lots of neurogenesis in the adult hippocampus, and then the pendulum sort of swung back, and then there were a few papers that said, no, we can't really see that. But actually recently, with the development of new techniques, including techniques such as taking advantage of a type of carbon dating, all the way from just sort of better imaging, better use of immunohistochemistry. And these techniques have actually really established that in the dentate gyrus of the hippocampus, there is actually abundant neurogenesis in the adult brain and a clear decline during the aging process. And some groups have now actually investigated postmortem brains from individuals who had Alzheimer's disease. And as it turns out, neurogenesis, this process of new neuron formation, it actually declines. It's almost completely lost in Alzheimer's disease. And it actually declines early on in disease onset. So even in individuals with only mild pathology have severe impairments in neurogenesis. And so given that these cells in this region of the brain are contributing to learning and memory functions, we really think that understanding why neurogenesis declines in Alzheimer's setting is important, and there is potential there for reversibility. So that sort of brought me to where I am now.
So could you say a little bit more about, we all think about, certainly if you think about neural development, you think about neurogenesis, during the very early development of the brain and nervous system. And then, as you said, when I was, I think just starting at Brown's, 1990s, there was a lot of discussion, highly controversial discussion about, do new neurons, are they born in the brain? I mean, I think just to make it very clear that the brain and, the spinal cord in particular is kind of an odd organ, right? It doesn't regenerate to the extent that other organs do or can. And so, and then this realization that there are these neural stem cells that you work on that do give birth to new neurons. How do you think about that? Do you think that that's occurring normally in the younger adult brain? Is the neurogenesis, the birth of new neurons, an ongoing process, or is it ramped up during under certain conditions? Like if we're thinking or remembering, do you need new neurons born to remember? Could you give the listeners a sense of what is neurogenesis? What is the typical thought about what that process is doing in the, maybe the mid-age, younger mid-age brain?
Yeah, absolutely. So first of all, most of what we know about adult neurogenesis comes from studies in rodents. So although it is, there's evidence for it in humans, we know much less about what it's doing functionally in humans. But I think we will get there. But in terms of what we know now, again, this comes from mostly rodent studies. And in rodents, there's abundant neurogenesis in this region of the hippocampus, the dentate gyrus. And these cells are mostly functioning in distinguishing between memories. And so this area of the hippocampus is important for sort of organizing memories, indexing memories, accessing memories. And so, a great example that is actually commonly used in the field is, okay, if you go out after work, you walk out into the parking lot and you need to find your car, and you have to remember where you parked your car today, not where you parked your car yesterday. And so 5 days out of the week, you're in this very similar context, and you need to be able to distinguish between those contexts. And that's just one example of a task that your brain actually has to do in different ways throughout the day. And so that's sort of where we're at in terms of functionally understanding these cells. And interestingly, so you touched on this question of whether we can actually stimulate neurogenesis. And it turns out there are a number of ways that neurogenesis can be stimulated at least in the rodent brain, and that can lead to better performance in cognitive tasks. So for example, exercise stimulates neurogenesis. It has to be voluntary exercise. If you force a mouse to run, it doesn't like that, and that won't help. Environmental enrichment. So you mentioned just thinking. So it has been shown, and the evidence is very strong, that environmental enrichment does increase new neuron formation and integration of new neurons. So absolutely use your brain and exercise. And there are other, there are plenty of other things like caloric restriction, modulation of TOR signaling, for example, a number of other ways that we know we can stimulate these cells as well. So there's hope.
Great, thanks. Well, thank you both for getting this, laying some foundation here, touched on a lot of different things. And I think what we're going to do is now, there's a lot of questions already coming in, and some of them are the questions we're about to ask you, so we might get fed from that. But Chris, I don't know, I think what would be really good is perhaps there's a question that came in the chat about, what is memory, what is dementia? And that was actually a prompt that I had to ask you, and I was going to ask both of you, can you describe what's going on in memory loss? Chris, maybe, did you want to jump in here, or should we just like what's recall? Is memory loss you're erasing a memory or your inability to recall? Steve, maybe you can take this and we'll get a little foundation here, and then Chris can pick up from there.
Well, this is a common question, so it's important. It's on people's minds that a lot of people are confused about the difference between Alzheimer's and dementia. And dementia is a term that means that there's cognitive impairment that interferes with day-to-day functioning. And it doesn't tell you the cause. There are many forms of dementia. Alzheimer's as a disease is the most common cause of it, but certainly head injury and Parkinson's and stroke and alcohol and HIV and 100 other things can cause dementia. What tends to happen there are different types of Alzheimer's, but different presentations of Alzheimer's, but the most common type is really what Ashley started to refer to as that involves episodic memory. So more recent memories, the formation and the storage of something that just happened recently, like where you parked your car, what you had for breakfast, that kind of thing, details of conversations that just happened or happened the day before. And the reason that's such a common symptom of Alzheimer's is that that area of the brain that Ashley's studying, the hippocampus, is involved with the formation of these new memories. And the tau protein, first amyloid protein builds up and later tau protein builds up, entangles inside nerve cells, and the connections start to break down. The tau protein spreads from one nerve cell to the next. The synapse, the connections start to break down, and eventually these new memories become less reliable. And people often get confused because someone can remember something from 50 years ago, but they can't remember what happened this morning. And that's very typical of Alzheimer's disease because those older memories are stored in other areas of the brain or in different packets that aren't affected early on typically. So usually Alzheimer's is a problem initially of short-term memory. Unfortunately, as the disease progresses, those older memories can get eroded as well because really the nerve cell loss becomes quite profound over time in Alzheimer's disease, unfortunately. And so even basic memories like recognizing your own family members can be eroded, and people lose that, which is tragic for families. Interestingly, and people are always very fascinated by this, certain procedural memories like playing the piano can be preserved for many years into Alzheimer's disease. That's a different type of memory that's stored, and sometimes people can even learn new things, learn new music, and remember that. They can't remember that they ever studied it or learning it, but they can actually perform it. And so it's amazing, and memory is not one uniform function, but what the real focus is here is short-term memory, which we need, like to find your car, to have continuity from one day to the next. If we're reading a book, we have to remember what we read the day before. That's a short-term memory storage formation issue, and that's vulnerable in Alzheimer's disease.
Yeah, I mean, one thing this puts me in the mind of is one of my mentors, Steve, was Sue Corkin.
Right. Perfect.
Yeah, she was and Sue's most famous for having worked with HM, the patient who had bilateral hippocampal damage, and I was able to meet him once. It's just amazing.
Yeah.
Incredibly fluid, super, one of the kindest people you'll ever meet, super engaged, and just from 5 minutes later, we'd get to the end of the story,
Sure.
and the end of the story would remind him of the beginning, and unless you broke the loop, he would tell the story over and over, because again, you get to the conclusion, and then that kind of brought him back to the beginning.
Right.
It's really startling how selective and profound. Yeah.
Her work with him proved the point about the distinction between procedural memory and
exactly
memory and declarative memory. Yeah.
Right. He was perfectly good at learning all sorts of routines, and it was really stunning. So one of the questions that we got from the participants, from the audience, I think really nicely dovetails with something I'm pretty curious about. So there's a really interesting connection between sleep and memory. And there's also an increasing connection between sleep and clearing out bad stuff in your brain at night, as the time when the garbage collectors come, as they say. Could you guys talk about the connection between sleep or brain clearance mechanisms maybe and memory formation as it relates to Alzheimer's? Is that something you could weigh in on?
You want to take the lead on that one? And
sure. I mean, I have two sort of very different answers that relate to my work in very different ways. And so in terms of the sleep, so it's well known that disturbances in sleep, circadian rhythms, other processes such as like body temperature regulation, as well as weight fluctuations occur early on in Alzheimer's disease. And so we've actually become interested in getting into a lesser studied region of the brain, which is the hypothalamus, which is regulating all of those processes. And so, there's, unlike the hippocampus, where there's lots and lots of work, and the importance is very, very clear. I think what's going on in the hippocampus is it's very murky and understudied. And so we are taking a combination of a genomics approach and a cellular reprogramming approach, actually, to try to look into this area of the brain and understand why it is that it's affected early on. And our hope is that if we can understand what's happening very early on in that area, it might actually give us some clues as to what's happening mechanistically overall in this disease, because it is a really early affected area. And in addition to that, hopefully that will actually lead to some strategies to treat some of these early symptoms of the disease. So that's the first part of your question. And then in terms of clearing out damage, that's actually something that we've been looking at in the neural stem cells. So now going back to the hippocampus, and actually another region of neurogenesis in the brain where we've looked, where rodents have a lot of neurogenesis, which is in the subventricular zone. And so what we find is that as neural stem cells activate and enter the cell cycle in order to form new neurons, they actually sort of flip a switch in terms of how they process cellular damage. And what that allows them to do when they flip that switch, they're able to actually clear out some of the damage and some of the aggregates that builds up in the cells over time. And so we haven't yet looked at the new neurons that have formed, but we're wondering now if those new neurons that have formed, do they actually have less damage than the older ones that have been hanging around for years and years? And that's sort of the key question now. Yeah, so two great questions, very different answers.
Yeah, I mean, there's a lot of interest in this area. And one of the risk factors for Alzheimer's is poor sleep. And and Ashley already mentioned a couple aspects of it, but another is in people who don't sleep as well, there's greater deposition of amyloid protein, of plaque type protein. And so understanding that mechanism and the clearance of sleep versus wakefulness and how to promote that is important. Another is, as Ashley mentioned about the hypothalamus, is that the circadian rhythm gets disturbed in Alzheimer's disease. And so the biological clock can be quite in disarray, which is very disturbing for family members and loved ones don't sleep as well, consequently, and their health is affected. So the sleep problem is really an important part of the Alzheimer's story, both mechanistically and practically on a day-to-day basis. So understanding it's important.
Yeah, thank you for that. One small connection that's kind of interesting here is one of the theories about sleep and clearance has to do with what's called the cerebrospinal fluid, the CSF, right? This fluid that flows all the way up and down. It's really interesting to think about, if it's actually acting as a clearance mechanism at night. That's one idea of sleep, right? Does that mean the CSF is just full of all of the bad things we want to get rid of? Is that useful? Is that interesting that this very particular kind of fluid in our brain has this role maybe?
Well, I think the fluid in the CSF spinal fluid is important because the protein aggregates and their other forms are deposited in spinal fluid. So that's a natural biomarker to measure changes. And so one thing we see in Alzheimer's disease, there's a characteristic signature in the spinal fluid that this toxic form of amyloid actually goes down in the spinal fluid as the plaques build up in the brain, and tau protein goes up, and phosphorylated tau protein goes up in spinal fluid as the tangles form in the brain as well. So it's a good way to measure activity of these proteins. It's a little problematic because we have to do a spinal tap or access the spinal fluid. And I think soon, looks like some of the biomarkers that were plasma biomarkers that are being developed sort of mimic this. So we should be able to get this peripherally without having to look at the spinal fluid. But another big aspect of the spinal fluid issue is that in late onset Alzheimer's, the most common form of Alzheimer's, it seems like there's decreased clearance of spinal fluid. And so there's a buildup of proteins and they're not cleared as well, as efficiently as they are in younger stages of life or in people without Alzheimer's. So that's one key physiologic aspect of mechanism underlying Alzheimer's. It looks like in younger onset Alzheimer's that is due to a genetic mutation, they have more trouble with overproduction of amyloid protein and tau protein. And clearance may be an issue, but it's not the primary issue. It's the production issue. But clearance is an important issue in later forms of Alzheimer's, later onset.
There's a question. I just wanted to kind of stay on the theme of neurogenesis just quickly, and then I think we're going to probably start to move into therapy and talking a little bit about some of your ideas about, where breakthrough therapies might come from. Why has it taken so long? So maybe I'll seed that as you start to think about this. But actually, there's a question just following up on discussion about neurogenesis in Alzheimer's disease patients, and asking whether if there's a decline in neurogenesis in Alzheimer's disease. Is that a consequence of or a causative of the disease? So is it a secondary or primary effect? Do you have thoughts about that?
Yeah, so we don't know, but I can speculate. What we do know, for example, is in animal models of Alzheimer's disease, we see the same thing as has been observed in the human brain. And so neurogenesis is down across animal models for the most part. We don't know to the extent that we will be able to ramp it back up by tapping into a more dormant pool of neural stem cells that we think is still there, and evidence suggests is still there. We do know that if you transplant cells into the hippocampus, transplant neural stem cells in, they can undergo healthy neurogenesis. If you transplant wild type cells into a Alzheimer's mouse, if you do the opposite, they don't do as well. So if you take the Alzheimer's cells and you put them into the wild type mouse, they don't make neurons as well. So we think that there is something intrinsic to these cells that is actually defective potentially, but we don't know the answer for sure to that. But we do think that the Alzheimer's environment is sort of permissive to neurogenesis. So if we can target the endogenous pool in an Alzheimer's individual, you would be able to increase neurogenesis, at least to some extent.
Mm-hmm. And is there a biomarker for neurogenesis, for active neurogenesis?
So unfortunately not right now in a living animal or person, but that would just be wonderful to have. But we currently don't have a great biomarker.
Right.
Yeah, but maybe Steve could show us the way on that one.
Right. I mean, that would be great.
Yeah, absolutely.
And then I have another really quick question, just maybe we'll wrap up the neurogenesis discussion. But let's come back to where memory's formed, and of course there's a distributed model But if you take, neuron dies, let's just imagine that there's some form of memory that's encoded at the cellular level, right? Okay. Synapses. A neuron dies, you add a new neuron. I'm just like, being really simple here. Put a new neuron in. Do you think that's sufficient to protect a memory? Is a neuron just gonna re-inhabit the location where the previous neuron was? It just disappears? Or is there a framework for new neurons to kind of embed themselves in and then form connections that are predefined? How do you think about the role of
potentially neurogen? I mean, again, I don't think we know the answer to this question, but I think potentially there is. I mean, I think there is potential for this, in part because in the dentate gyrus in particular, these new neurons, they don't have to go very far, right? So the neural stem cells actually only migrate as they mature. The cell bodies only migrate a couple of sort of cellular layers away. So we're not talking about like in the rodent olfactory bulb in this subventricular zone, for example, where the cells have to get all the way from sort of the middle, almost the middle of the brain up into the olfactory bulb. We're talking about, microns. So it's a very small distance that the cells have to go. And, I think that there is enough circuitry in place for these cells to integrate properly, at least to some extent. Of course, the further a person is along in the disease, the more poorly this process is going to work, I would anticipate. But potentially, in combination, for example, with some of the strategies that Steve has been working on, we could actually make this work. If we improved certain functions, like reduce the inflammation of the brain, for example, I think that would really provide a setting for, better integration of these cells.
And of course, we've been very neuro-focused here, Chris. And actually, thinking beyond neurons.
There are questions coming in that I'm really gratified about, because it's what I've really wanted to ask you about as well. So again, a really good synergy here. So one of the questions is, can you speak to the roles and pathology of cells other than neurons, e.g. microglia, astrocytes, other kinds of cells? I mean, I'm particularly interested maybe to turn this towards the question of therapies and treatment. Are there great discoveries lurking there that are gonna maybe help with treatment, or are there treatment avenues that can be uncovered if we sort of go beyond the neurons as targets or as complementary forms?
Well, I can start in, and then Ashley, you can weigh in about this. Well, I think Ashley said it right early on. I mean, the aging is the biggest risk factor for late onset Alzheimer's disease. And so understanding how neurons age, the neuronal environment, what supports them, what promotes viability of neurons with aging, how to maximize that, optimize that is critical. And I think one thing we've learned, key thing we've learned over the last 25 years in Alzheimer's research is really to have an open mind and not take a monolithic approach to this, that really try to understand aging. And so neurons are important, the vascular supply and changes of blood flow and blood vessels are important, glia are important. And, there are many components. And I think some of the key elements, and that's why I'm so excited what's happening here at Brown at the moment, is really looking at the plaque and tangle type of pathology that we're aware of, but really looking at geroscience, looking at genetics, and really seeing what the components are and what can be modified, and generating new treatment targets. So I think we're on the verge of, Ashley's already cooking up stuff that is promising, and I already mentioned about John Sedovy and others that there already are emerging targets that hopefully will be have clinical benefit.
Yeah, I mean, I would agree with that. And just circling back to the undergraduates who've been developing a cure for ALS. I mean, so many of these hallmarks of aging that we've been studying, such as mitochondrial dysfunction, for example, these are areas where I think, treatments are being developed and considered as well. And it may be that that what we need is some sort of combination therapy to both clear the plaques, as well as enhance neurogenesis to get the dentate going again, as well as reduce the oxidative stress and the neuroinflammation. And that's sort of where these former Brown undergraduates sort of, the light bulb went off, and they're using a combination therapy that seems to be effective for degenerative disease.
So we moved into treatment, and I think there's a question here, but it's, I think, the question on all of our minds. Maybe we'll start with like why so slow? I think that this is always the question. Probably Steve gets asked fairly frequently. But why is it taking so long, Steve, to find even something to slow the degeneration of neurons?
Right.
Not even a cure. Just if you slowed it you decrease the rate of decline. Big improvement over 30 years. That's hugely significant in this area. So.
But you have to be both patient and persistent at the same time. So it's a tough balancing act. I think, Alzheimer's is complicated. It's wrapped up in aging, and neurons are sort of built for, there is neurogenesis. I agree with Ashley, but by and large, you have your full complement of neurons, and you got to take them all the way to the house. And if you're at increased risk for Alzheimer's, for genetically or for other reasons, then you're working, against the odds and time. I can see the future. Mm-hmm. I can't say specifically what the answer will be, other than I see a combination of treatments for Alzheimer's. I take heart from, no pun intended, from HIV. There was initially no treatment for HIV, and then we had a first drug that had some mild benefit, but was limited. And then through sheer insistence from the patient community, and Dr. Fauci was the focal point at the time, so he's been through a few of these wrangles in his lifetime, in his career. They insisted that better treatments come up, and combination treatment now has a big impact for HIV. And so I think Alzheimer's is going to require a combination of treatment. I don't think there's going to be one drug, one amyloid-lowering drug, or a tau blocker, or a mitochondrial protector, or something like that alone that is going to be effective. And then there are different, subtypes of Alzheimer's, and we're going to have to personalize it and make it precise depending on the pathology that people have. So we're close. We've got drugs now that, and I'm sure the audience has followed along recently with one of the antibodies called aducanumab that is being developed by Biogen up in Cambridge, that does substantially lower amyloid plaques. And there's been some encouraging data. It's a little problematic because the phase 3 trials ended early, so the data set's incomplete. But there are 2 out of 3 trials showed positive clinical benefit. So it's the first time we've actually seen that an amyloid-lowering drug had some clinical benefit. So I think it's the beginning of this therapeutic breakthrough. I think it's the very beginning, and we really need to build on that to find other treatments that will, I don't know whether this drug will be approved. The FDA is considering it right now, but if it is, we need to build on. That's just the stepping stone to build on. Hopefully work here at Brown will contribute to that. We'll be part of that combination package. And
I totally agree with Steve. And I would just add that Another reason that I think progress has been so slow is because the tools that we have in hand to study this disease have been limited. So at this point, the field recently has been moving into new model systems such as IPS and cellular reprogramming model systems to study this disease in a dish. And 15 years ago, we didn't have anything like that. So it wasn't possible to actually make patient-derived neurons. And the field can now do this, and we can study them in a dish and do things like screening, drug screening, small molecule screening in a dish. And in addition, if we combine those reprogramming tools with gene editing and genome editing tools, CRISPR-Cas9 based tools, then we can really start to study mechanism better than we could, several years ago, even 5 or 6 years ago. And now there's sort of a new wave in the field where people are starting to use organoids and get away from 2-dimensional cultures and getting into, modeling the disease in 3-dimensional cultures where we can actually see how the cells interact. Because as Chris and Steve alluded to earlier, it's not just neurons, right? I mean, these cells are interacting with glia, microglia. There's just so much more going on in the brain than just what's happening in the neurons. And so I think that these tools are really going to accelerate progress, and I'm very hopeful that that we're not.
I totally agree with that, because one of the big problems with the transgenic models, the mice models we've been using, is they replicate one form of Alzheimer's, one component, the amyloid buildup, very well, but they don't have the neurodegeneration and the tau change and the inflammation and the vascular change. So they're not really it's not really Alzheimer's disease. It's one element of Alzheimer's, and you can treat that element, but you don't necessarily rescue the disease. So having these models that are more complex, that better replicated, is very important. And then I guess the key point I'm sure is on people's minds. I think just common sense that the earlier we intervene in a neurodegenerative process, the better. So catching the brain while it's still healthy, and hopefully someday, even before amyloid plaques, the whole process has begun that we can detect, and preserving that healthy neuronal environment is critical. So starting early before a lot of damage is done. I think there are things that we all can do, and Ashley already referred to a couple of them, are lifestyle changes. So, exercise, a healthy diet, Mediterranean type diet, brain stimulation within our activities every day, being engaged with other people, good heart health, are going to keep the brain healthy, it's going to keep the heart healthy, and it's going to lower the rate. That's something all of us can do, and starting early in life. To prevent dementia and other bad things from happening later in life, because we want to make it to the finish line in good shape. And so that's something we all can do. The other thing I take real heart from and encouragement is a gene, and Ashley started mentioning them, as our gene-based treatments. I think some of these are going to be powerful for neurodegenerative disorders like Alzheimer's. So we've seen in spinal muscular atrophy, which is a terrible disease of childhood, which had no treatment, and children died at a young age, were very disabled. And now we have gene modifying treatments that actually block the toxic proteins, or replacing the gene that's missing, the abnormal gene. And the children do way better. So we've had this is really what, Diane, you were referring to. We've had the impact in that disease already, which is what we're looking for in Alzheimer's, because it's substantial. It's not just, borderline, you can debatable, but it's really substantial. And we're starting to use some of those same treatments in Huntington's disease, in ALS, and in Alzheimer's disease. So early next year, we're going to be starting some tau-based gene modifying treatments. And I think it's going to have legs eventually.
Yeah, I mean, I think this is certainly, of course, a really encouraging direction for the hereditary forms of neurological disorders, where you have a clear target, and there is certainly hope that those treatments might have some translation to the less well-defined, non-hereditary, more sporadic disorders that of which there are many, including the Alzheimer's diseases. So, you know, it is, you have fewer parameters to deal with when you know the gene and
right.
there's a clear mutation. And I think that is definitely optimistic. One of the things that was mentioned, you know, from someone we know well, who said, of course, that this is well known to you, Steve, but in terms of why so long, you know, that you take an action and then the outcome data is just many, many, many years away, right? And then the ability to assess outcome, you know, what are the diagnostics that one is looking for when one is assessing treatment. And so there's this built-in incredibly long time scale over which one is kind of assessing any form of treatment. And so
this is actually a huge
incredibly
issue.
difficult. Yeah.
This is a huge issue, Diane, in Alzheimer's, especially if we go earlier in the disease. So someone might be building up amyloid plaques, but is cognitively normal, has no memory loss. And let's say we want to test a treatment. We want to prevent lower amyloid buildup. The rate of change before they get memory loss is in years. And so we have to recruit a group of people over a number of years, and then we have to follow them for a number of years. So we don't have good prognostic biomarkers yet, or theragnostic, to tell whether or not the treatment we're using is going to work. That gives us, like, for example, if you have a cancer drug that reduces the tumor size by 50% in 6 months, you can get a readout. You know, either this drug is going to work or not going to work for that particular patient. We're talking, like you said, years, and especially the earlier you go, the longer it takes. So you don't know if we need to get better predictive measures so that we don't devote that whole time. We don't devote 5 or 10 years before we find out we're on the wrong track. It's not very efficient, right? Yeah. So that's critical. So the biomarkers are critical.
Yeah. So Chris, do you want to, we have like 3 minutes to the hour, and as we've always said, but really honestly, I think I could sit here and talk to you guys for several more hours. And there's just been such a great set of questions coming in. We couldn't get to them all. But Chris, do you have a final question before I do a little bit of a wrap up here?
No, not in particular, other than what's the phenomenon where you don't notice time passing and then suddenly it's an hour late? Is that an early warning sign of a decline of some kind?
I mean, maybe the question then, if you can both answer it in 1 minute each, I don't know if this is possible. Quite rather a cruel way to to thank you, but you know, where do you, you know, if you were, where do you think, you know, what is your best hope for treatment? And I think we all, I would say, have a lot of optimism here because of the funding, because of young people coming into the field. And I'll do a little bit more of a wrap up here, but I really think there's just tremendous interest now, and I think that's so incredibly important. But what best guess, like what would you, mechanism, target?
Mechanism. I mean, I think one minute. I mean, early intervention is key. I don't think it's going to be one target, and I think it's going to come down to personalized medicine, where eventually we will know most people's mutations, and it will be different mechanisms in different individuals. And I think it'll take some time to sort of go down the list, and it may be that everybody gets something to help with the pathology. Maybe everybody gets someone to keep the plaques and tangles down, but then they get something else on top of that, you know, whether their issue is more in their microglia, for example, or if it's oxidative stress at astrocytes or neurons. So that's my best guess, which I know isn't very specific, but
right.
We're not there yet, unfortunately.
Yeah, there's a lot of agreement here. So I think healthy lifestyle, that's incumbent upon all of us, as something we can do to keep our hearts and our brain healthy. I think combination treatment, and it will vary, as Ashley said, depending on what the pathology is for an individual or what the risks are, starting early. Talented young investigators from all different fields, get more perspectives and more techniques, and then some things I can predict, that, like Ashley said, there will be the targets we already know, amyloid and tau and inflammation and so forth. Things are going to come out of left field. There's going to be discoveries that we had not anticipated, and some are going to be genetic, as I mentioned, but some are going to come out of like what happened with the Brown undergrads, that they came up with their own potion that no one else had predicted. Undergrads came up with this, and that's going to be great. And that's why we have to keep the open mind, because we may not know that target or that combination yet. And that's where the creativity comes in. It's going to be a lot of fun.
Well, thank you both. So I'm going to do a really quick wrap up, and then I'm going to mention the next Carney Conversations. But so we heard Alzheimer's disease is not a single disease. There are likely to be multiple origins, and there certainly are. That means that there's likely to be the need for a combination of treatment. I think one thing that's close to Chris's heart that he constantly tells us, as we have lots of discussions about, the brain as a system of systems. It isn't just neurons. And many of you put comments into your questions. Put comments in about the fact that, of course, there are glial cells, and microglia, and they outnumber neurons by manyfold. So there are many possibilities in terms of thinking about new targets, but it's going to take a concerted effort, and it's going to take a real integration of people coming into the field from a number of different perspectives. And I think that's really happening. And I think that there's a lot of reason to be incredibly hopeful right now. The path is through research. I mean, understanding mechanism. When you've got a mechanism, you have targets, and that's just that has to be the route. And I think that there is no dispute about that. But I think there hasn't been this real influx of people with a lot of different perspectives coming into the field until the last few years. And I think that's been really tremendous, including Ashley, and all of her experience in neurogenesis. And, I think Steve's incredible ongoing optimism, but treatment, thinking about early diagnoses, right? Steve, you mentioned that, and Ashley mentioned it, and Chris and I've talked about this a lot. Like, what are you looking for? And the earlier you can get some kind of diagnostic, the better. And so definitely lots of reason to be very hopeful, but also a lot of people out there who are really desperate for treatments and cures, as we are. So that's my wrap up. I wanted to thank you both just hugely for being here. It was incredibly interesting, important, really important area, and I think we opened more discussion than we ever possibly imagined. And so we're going to take this back to the lab. I don't know why we're here. We should be in the lab.
Got to get the research. Got to get the work.
Yeah, right. I just wanted to also mention that the next Carney Conversations is going to be on December 15th with David Better, who's a professor of cognitive linguistic and psychological sciences. And he just published a new book called On Task. And so we hope you'll join us then for that conversation. In the meanwhile, thank you so much for being here. Thank you for the incredible discussion. We barely got to, but really very stimulating. Steve, Ashley, Chris, bye. Thanks.
Thank you so much.